Welcome to the Deep Dive. We're the place you come to get beneath the surface of the latest research.
Unpacking the key insights.
Exactly. And today, we're jumping into something pretty exciting. Medical image classification. It's a field with huge potential for AI.
Massive potential, but also some really unique challenges.
Right. And specifically, we're looking at how a newer approach using graph neural networks, GNNs, is tackling what you might call the messy data problem, something traditional AI, like CNNs, can sometimes struggle with.
It's a really important distinction. Convolutional neural networks, CNNs, they have been the bedrock for a lot of AI progress. They're fantastic when your data fits neatly into a grid. Think standard photos.
Yeah, very predictable structures, but medical images, they're often anything but neat, aren't they?
Not at all. You're looking at, say, histology slides with complex cell arrangements or intricate retinal scans, maybe tissue morphology where features are, well, irregular, they overlap.
And they have really important structural relationships.
Precisely. And if you try to force all that rich structural information onto a simple grid, like a CNN expects, well, you risk losing the crucial context.
You lose how things relate, like how cells group together or where a lesion sits relative to other tissue.
Exactly that. And often, that relational context is the key. The defining feature of a disease might not just be that an abnormal cell is present, but maybe its specific closeness to a blood vessel or how it connects to surrounding tissue.
Okay, so that's where GNNs come in, because they're designed for relationships.
They're inherently built to model relationships, to understand connections within data. So yeah, for this kind of problem, they often have a big advantage over just counting pixels or simple features in a grid.
Makes sense. So our source material today zeroes in on this specific framework called iPac. That's I-P-A-C.
Stands for Incorporating Intra-Image Patch Context into GNNs. Quite a mouthful, but the core idea is powerful.
And the mission here, as I understand it, is basically to ditch that rigid grid structure.
Right. And instead build a kind of semantic map, one that really captures the underlying relationships between different visual elements in the medical image.
And the payoff seems significant. The research suggests iPac improves accuracy by up to 5% on average compared to other GNN methods.
That's what they found, yes. And in medical diagnostics, a 5% improvement isn't trivial. It can be a really substantial gain in clinical precision. The versatility of iPac is a big part of its appeal, too. It's designed as a more general framework.
Okay, before we unpack the iPac method itself, maybe let's quickly touch on why GNNs were needed in the first place here. We know CNNs struggle with the structural stuff. What did earlier attempts using GNNs look like? What was the landscape before iPac?
Well, people were definitely trying to apply GNNs. Some early approaches involved trying to force the image into a graph structure using techniques like superpixel over segmentation.
Superpixels, like grouping similar-looking pixels together first.
Yeah, basically breaking the image into visually coherent blobs, then treating those as nodes. Or using more complex region proposal methods to identify potential areas of interest.
Okay.
We also saw things like hierarchical GNNs being used, maybe for spotting lesions in mammograms, or attention-guided GNNs trying to analyze Alzheimer's disease progression from MRI data.
Right. Those sound quite specific, though, tailored for one type of image or task.
Often, yes. And that's where iPac aims to be different. Its novelty is really in its generic approach. It avoids relying on these potentially complex, sometimes domain-specific segmentation steps.
So how does it create the graph, then?
It converts the image into a graph by focusing on relationships between different clusters of image patches. It's a higher level, more semantic way of representing the image structure.
Clusters of patches, okay.
And that makes it potentially much more versatile, applicable across different types of medical images, whether it's skin lesions or retinal scans or pathology slides.
Okay, the theory clicks. But the practical implementation, how do you actually go from a flat, static image file to this dynamic graph of relationships? Let's walk through the iPac process.
Yeah, let's do that. It breaks down quite neatly into 3 main stages.
Stage 1 seems to be about getting the raw materials ready, patching and feature encoding.
Exactly. First step, pretty straightforward.
Okay.
The image just gets divided up into a fixed grid of smaller squares or patches. Think of it like tiling the image.
Simple enough, but then you need to represent these patches somehow, right? Not just as raw pixels.
Correct. You need meaningful representations. And for this, they use an autoencoder, specifically one based on the SWIN transformer architecture.
Ah, the SWIN transformer. We hear about that a lot in Vision AI. Why that specific choice here? What makes it suitable?
Its strength really lies in handling visual information at different scales simultaneously. That's crucial for medical images. Well, think about it. Sometimes the diagnosis might depend on the really fine-grained texture within a single cell. Other times, it's about the overall pattern of damage across a much larger area of tissue.
Right, micro and macro views.
Precisely. The Swin transformer is good at capturing both. So the autoencoder takes these patches and compresses them into these dense, high-dimensional vectors embeddings that retain both that fine spatial detail and the broader semantic meaning.
And they make sure the encoding is accurate using mean square error, comparing input and output.
Yep, that's the standard way to train the autoencoder effectively. Ensure it learns to represent the patches faithfully.
Okay, patches encoded. Now, stage 2. Clustering and graph construction. This sounds like where the graph actually takes shape.
This is where it gets really interesting, yeah. So you have all these high-dimensional embeddings, one for each patch, potentially hundreds, even thousands of them. There's probably too many individual nodes for an efficient graph.
Right, would be too complex.
So what they do is they cluster these embeddings. They feed them into a standard, off-the-shelf clustering algorithm, something like k-means.
K-means, so just grouping similar catch embeddings together.
Exactly. The goal is to automatically group patches that are visually or semantically similar. It's a form of data reduction, but it also helps discover higher-level structures in the image.
So instead of a node for every tiny patch, you end up with nodes representing what, like different types of tissue or structures?
Precisely. You might end up with, say, 50 or 100 nodes, and each node now represents a detected cluster of patches. Maybe one cluster represents healthy tissue, another represents inflamed tissue, another a specific type of lesion, and so on. These clusters become the nodes in our graph.
Okay, nodes defined. What about the connections, the edges between these cluster nodes? How are they decided?
This is key for capturing the spatial context. The edges are built based on how often patches from different clusters are actually neighbors in the original image grid.
Ah, so it's about spatial adjacency.
Yes, but measured quantitatively. If patches belonging to cluster A are frequently adjacent to patches from cluster B in the original patch grid, then the edge connecting the node for cluster A and the node for cluster B gets a strong weight. Let's call it A.
So the more neighboring patches between two cluster types, the stronger the connection in the graph.
Exactly. This way, the graph encodes meaningful spatial relationships and the density of connections between these discovered semantic entities within the image. It's not just if they're connected, but how strongly or how often.
Right, it becomes a map of significant interactions. Which brings us nicely to stage 3, graph-based learning and classification. We have the graph, now what?
Now we leverage that graph structure using a GNN. The specific technique highlighted in the sources is edge graph convolution.
Edge graph convolution. Okay, that sounds a bit more advanced than a standard GNN layer. How does it differ? What's the edge part doing?
Good question. So many standard GNNs, like basic graph convolutional networks or GCNs, focus primarily on the features of the nodes and just the fact that an edge exists between them.
Okay.
Edge graph convolution takes it a step further. It explicitly considers not just the node information, our cluster representations, but also features associated with the edge itself.
And in iPac's case, that edge feature is that adjacency strength we just talked about, the weight A.
Exactly. The model doesn't just know that cluster A is linked to cluster B, it also considers how strongly they are linked, based on that spatial adjacency frequency. This allows the GNN to capture more fine-grained structural information during its learning process.
I see. So the message passing between nodes is modulated by how strong their connection is. It adds another layer of relational detail.
Precisely. This refined information then propagates across the graph as nodes update their representations based on their neighbors. You end up encoding both local details from the original patch features embedded in the clusters and the global context from how these clusters interact across the image.
That combination sounds powerful for clinical accuracy.
It aims to be yes. Capturing both levels is often critical. The final step is usually a standard, fully connected layer with softmax activation to make the actual classification prediction based on the final graph representation.
Okay, methodology covered. Let's talk results. How did iPac actually perform when they tested it? They used 4 BreastMNIST 2D datasets, right? Pathology, skin, breast, and retina.
That's correct. 4 diverse datasets to test its versatility. And the results were pretty consistently positive.
How positive?
Well, iPac consistently did better than the baseline GNN method they compared against the one using superpixels. For instance, on PathMNIST, the pathology data, iPac reached 91.4% accuracy, whereas the baseline was at 90%.
Okay, an improvement. What about against other types of state-of-the-art models, maybe non-GNN ones?
It held its own very well there too, and sometimes came out ahead. A notable example was on the BreastMNIST dataset. There, iPac achieved an AUC area under the curve, a common performance metric that was 9.4% higher than a model called MedMamba.
Wow, 9.4% is a big gap. That suggests it's really good at picking up subtle signs in those images.
It seems particularly strong at leveraging those structural diagnostic markers, yes. It's worth noting, though, that one dataset, RetinaMNIST, the retinal scans, proved the most difficult for all methods, including iPac.
Lower overall accuracy there.
Yes, it suggests that even with these advanced graph methods, dealing with extremely fine-grained structures like the dense vessel networks in retinas remains a significant challenge.
Fair enough. Now, the researchers also did a lot of tuning and testing, didn't they? Ablation studies to figure out what architectural choices actually matter most for iPac.
They did, and there are some really valuable takeaways there for anyone building similar systems.
What stood out? Like, which GNN layer type worked best?
When they compared several common GNN layers, standard GCN, GIN, GAN-T, they found that GraphSage, specifically the SageComb implementation, consistently gave the best average accuracy across the datasets.
GraphSage, why might that be? For listeners maybe not deep in GNN architectures, what makes GraphSage different or potentially better here?
Well, without getting too technical, GraphSage, which stands for Graph Sample and Aggregate, is particularly known for its ability to generalize well. It's designed to learn how to aggregate information from a node's local neighborhood in a way that can be applied even to nodes or graphs it hasn't seen during training.
Ah, so it's better at handling new, unseen data, like images from new patients.
That's the idea. In a clinical setting, you absolutely need your model to be robust and perform well on new cases. GraphSage's inductive capability, its ability to generalize, likely makes it a very suitable choice for this kind of medical application compared to some other layers that might be more tied to the specific graph structure seen during training.
That makes a lot of sense for real-world use. Now, you mentioned findings about complexity, too, something about not making the model too complicated.
Yes, that was a clear finding. It's almost a classic machine learning lesson. More complex isn't always better.
What did they see?
They found that increasing things like the internal size, the dimensionality of the GNN layers, or adding more layers in the multi-layer perceptrons used within the GNN, it generally led to a drop in accuracy.
Really? Why would making it seemingly more powerful hurt performance?
Overfitting, most likely. A more complex model has more capacity to essentially memorize the training data, including its noise and specific quirks. That means it might perform brilliantly on the data it trained on, but poorly when shown new real-world examples.
So keeping the GNN architecture relatively lean and focused on the core structural information learned from the graph was actually better.
It seems so. Simplicity, or perhaps appropriate complexity, won out. Maintaining that clear structural understanding was more effective than just throwing more parameters at the problem.
Interesting. And what about optimizing the graph structure itself, like the number of clusters?
That provided a really fascinating insight, actually. It wasn't a one-size-fits-all answer.
Oh.
For several data sets, including BreastMNIST, they found that using a lower number of clusters, maybe only 12 or 24, yielded the best results.
Fewer clusters. What does that imply?
It suggests that for those particular tasks, the most important diagnostic information might be captured by relatively high-level, broader semantic groupings in the image. You don't need extreme granularity.
But that wasn't true everywhere.
No. Remember RetinaMNIST, the really challenging one?
Yeah, the retinal scans?
For that data set, the optimal setup was the opposite. They needed more clusters, so higher granularity combined with smaller patch sizes to really capture the necessary details in those fine vessel structures.
Wow. So the complexity of the underlying image data really dictates how fine-grained your graph representation needs to be.
Absolutely. It highlights that while iPac provides a general framework, the optimal hyperparameters like cluster count and patch size are definitely dataset dependent. You need to tune it based on the specific visual characteristics you're trying to analyze.
But they did offer some general guidance, right? A starting point.
They did. Based on their experiments, a good general starting point seems to be aiming for a graph with around 50 to 100 clusters and using patch sizes somewhere between 32 by 32 and 96 by 96 pixels.
Okay.
That range seems to provide a reasonable balance between capturing sufficient detail, maintaining computational efficiency, and achieving good performance across typical medical imaging tasks.
That's very useful practical advice. So let's pull back a bit. What's the big picture takeaway here? What does iPac signify for medical AI?
I think it strongly reinforces the idea that for many complex medical imaging tasks, moving beyond just analyzing pixels in isolation is crucial. Focusing on the structure, the relationships, the context, that's where the next level of accuracy and potentially diagnostic insight lies.
And iPac provides a solid new baseline for doing that with GNNs.
Exactly. By representing the image through these semantic clusters as nodes and connecting them based on spatial adjacency, it finds an effective way to integrate both the fine-grained local features captured by the patches and the higher-level global context revealed by how these clusters interact.
It's kind of teaching the AI to see the forest and the trees in a way. Or maybe how the different kinds of trees are arranged in the forest.
That's a good analogy, yeah.
Right.
It helps the AI reason more like, say, a pathologist who doesn't just identify individual cells, but understands their organization within the tissue architecture.
Which leads us to a final thought, maybe looking ahead. The current iPac uses pretty standard components, right? K-means for clustering, spatial adjacency for edges.
Relatively off-the-shelf building blocks.
Given how sensitive the results were to things like the number of clusters varying drastically between datasets, what happens if you make those components smarter?
That's the really exciting next question, isn't it? What if, instead of fixed k-means, the model could learn or adapt the clustering approach itself? Or perhaps even more powerfully,
Yes.
what if you integrated more sophisticated attention mechanisms into the graph construction or the edge definition? Imagine the GNN could learn to dynamically weigh the importance of different relationships.
So it wouldn't just see that cluster A is next to cluster B, but it could learn that this specific type of adjacency in this specific context is highly predictive of disease, while other adjacencies are just noise.
Precisely. If the AI could learn not just the structure, but the relative importance of different parts of that structure dynamically based on the diagnostic task, that could unlock another level of analytical power.
Moving from just describing the structure to understanding its functional or diagnostic significance.
That feels like the next frontier for this kind of structural analysis in medical imaging, making the relationship mapping itself more intelligent and adaptive.
A fascinating direction. If the AI can learn the most critical relationships on its own, that truly moves towards more autonomous diagnostic capability.
It's a challenging but very promising path forward.
Well, this has been a fantastic deep dive into a really cutting-edge area. Thank you so much for walking us through the intricacies of iPac and GNNs in medical imaging.
It was my pleasure. It's exciting stuff.
And thank you, our listeners, for joining us on the deep dive. We'll catch you next time.